Hello everyone and welcome back. The nervous system will be the final topic that we will cover in the Anatomy and Physiology 1 lab. We will have two subtopics within the nervous system, the first of which is the brain, which will be covered during this lab presentation, and our final lab recording will cover the spinal cord and the reflexes associated with it. In your lecture class, you are likely to cover the functions of each of the individual parts of the brain, but here in the lab class, we want to give our focus to identifying the anatomical structures that can be found in the brain specifically. But before we start to go over the anatomy of the brain, we first need to review the two types of cells that can be found in the nervous system. Making up the nervous system, we have neurons and neuroglia. Neurons are the fast communicative cells that our body has. Each neuron is going to have its own dendrites and one long axon that's going to project from the cell body. The dendrites are responsible for bringing information into the cell body, and the axon is responsible for sending it out. The axon can be found wrapped up in what is known as the myelin sheath, which are these yellow things that you can see along the axon. Their purpose is to insulate the axon to increase transmission speed of action potentials from the cell body down to the axon terminals. It is thanks to the myelin sheath that our axon will have very fast signal transmission. There are several different structural classifications of neurons, but we are looking at a multipolar neuron since there are multiple projections that can be found coming off of the cell body. You can see another histological section of a multipolar neuron right here. We've got the very long axon and the many dendrites that will be coming into the cell body of our neuron. Now around the neuron we'll find many other glial cells, and these glial cells are supportive cells that are going to support the neurons in different ways. The myelin sheath, for example, is made up of different glial cells wrapping around the axon. We have many different types of glial cells, and I expect that you'll cover those in more detail in your lecture class. But you'll find all these glial cells scattered around the neurons, ultimately supporting its communicative function. So now that we've just briefly introduced the two types of cells that can be found in the nervous system, it's time to start breaking down the principal components of the brain. The brain's anatomy can be separated into four principal regions. We have the cerebrum, which can be shown up here. We have the cerebellum, which can be found back here. We'll have the brainstem that will be found down here, stemming out from the brain. And then we have a fourth principal region known as the diencephalon, which is not shown on this slide, but will be viewed shortly in a moment. Each of these principal parts will be made up of various other structures, and each structure will serve its own function for the brain. But again here, in the lab class, we need to focus on the anatomy of the brain and its overall organization. So we're first going to break down the cerebrum. The cerebrum is the largest of the four principal parts of the brain. We're seeing this brain in the sagittal section, and we'll note that the cerebrum contains the cerebral cortex, the corpus callosum, as well as the lateral ventricles. These are all structures that we will point out very shortly. The first thing that we do need to understand about the cerebrum is that it will be divided into two different hemispheres by what's known as the longitudinal fissure. You can see this is our left hemisphere and this is our right hemisphere. Each of these just refer to a different half of the cerebrum. What's quite interesting about the brain is that each hemisphere is going to control the opposite side of the body. So information from the right side of the body will actually come up to be processed in the left hemisphere of the cerebrum, and vice versa with the right hemisphere of the cerebrum as well. The cerebellum, which you can see down here and will be described shortly, will also have a right and left hemisphere. This slide shows us an inferior view of the brain, and you can see the cerebrum shown right here. This will be our left cerebral hemisphere, and our right cerebral hemisphere can be found over here, separated by this longitudinal fissure. You'll see the cerebellum shown down here in its two hemispheres, as well as the brainstem, which can be found stemming from the brain right here. The cerebrum can be separated into 5 different lobes, and each lobe is going to offer the body a specific function. First off, we'll have the frontal lobe, which can be found right here. We'll have the parietal lobe, which can be found right here, shown in blue. The occipital lobe will be shown in green and be responsible for processing vision information. And then we'll have the temporal lobe, which can be found out here. Those are the 4 major lobes, but if you go a little deeper, you'll also find the insular cortex, which can be found deep to the other brain lobes. There is quite a bit of cerebral anatomy, but one structure in particular that I want to point out is known as the central sulcus. The central sulcus is an invagination that will separate the frontal lobe from the parietal lobe. And when you get to the Anatomy and Physiology 2 lab class, you're going to talk a little bit more about this central sulcus. Each of these different lobes of the brain is going to offer a different function to the brain overall. I've already mentioned that the occipital lobe is processing vision information, but we'll see that the parietal lobe will process sensory information, and the frontal lobe will primarily be responsible for regulating motor information. Among the many things that the temporal lobe does for us, hearing information will come into the temporal lobe to be processed. You can see on this diagram we have our frontal lobe shown here, separated from the parietal lobe by that central sulcus. It's a very heavy invagination that will be separating these two lobes, and that's how we can tell the difference between those two parts in particular. We'll find the temporal lobe of the brain over here, and we'll find our occipital lobe towards the backside of the brain. So coursing all over the cerebrum, we'll find gyri and sulci. The cerebrum in general is covered with different gyri and sulci. Gyri in particular are these outward foldings that project from the brain, and sulci are invaginations that fold inwards. Gyri can be described as hills, and sulci can be described as valleys. Sulci can be considered different sizes. However, when they're very, very large, we call them fissures. This would be an example of the longitudinal fissure that separates our two cerebral hemispheres. Essentially, a fissure is just a very, very deep sulcus. Oftentimes, gyri are called hills and sulci are called valleys. One other distinction that we need to make about the cerebrum is that we're going to have gray matter and white matter. Gray matter can be seen wrapping around the outside of the brain shown here, and white matter will be found deep to the layer of gray matter wrapping around the outside. Gray matter is literally made up of neuronal cell bodies, and white matter is literally made up of myelinated axons that project from those neuronal cell bodies. So we'll see our neuronal cell bodies coursing around the outside of the brain, and we'll see their myelinated axons projecting into the white matter. The layer of gray matter along the outside of the brain is known as the cerebral cortex. And our cerebral cortex is one of the most highest order parts of the brain that we have. You can really see the gyri shown over here and all of the sulci that can be found in between the gyri. You've got your longitudinal fissure shown here, which is just a very, very deep sulcus. We'll also find a transverse fissure that can be found between the cerebrum and the cerebellum as well. You'll also see on this slide the ventricular system that can be found housed within the brain. Ventricles are spaces that can be found inside of the brain containing what's known as cerebrospinal fluid. Ventricles are lined by a special type of neuroglial cell known as the ependymal cell. And all of our ventricles are going to be connected to each other in some fashion to allow for the communication of cerebrospinal fluid from chamber to chamber. There are a total of 4 ventricles that can be found on the brain. Up here in the cerebrum, we'll have 2 lateral ventricles that can be seen and look rather large. Those will both be connected with the 3rd ventricle, which is housed between another principal region of the brain known as the diencephalon. The diencephalon will be pointed out shortly. The 3rd ventricle will be found connected to the 4th ventricle through what is known as the cerebral aqueduct. And the 4th ventricle is going to be housed within the brainstem. As the brainstem will eventually project off the spinal cord, the central canal of the spinal cord will course down here and also be continuous with the ventricular system of the brain. The 1st and 2nd ventricles are also known as the lateral ventricles. And neither the right nor the left ventricles will be assigned ventricle 1 or 2. We collectively say that the lateral ventricles are ventricles 1 and 2, and we have the 3rd ventricle found here in the diencephalon, and then we'll have our 4th ventricle found housed within the brainstem. So once again, you can see the cerebrum shown here. You have your cerebellum, which can be found here towards the backside and the base of the brain, and then you've got the brainstem, which can be found pointed out right here. From the brainstem, the spinal cord will project and travel down the vertebral canal. But we also have this 4th principal region of the brain known as the diencephalon. And this structure can be found here most centrally located in the brain. The diencephalon in particular is composed of 3 different structures. We'll find the thalamus, which can be found right here, the hypothalamus, and also the epithalamus, which is not labeled on this slide. The epithalamus will be labeled on another slide very shortly. The cerebrum does have some other subparts to it, but the cerebral cortex is going to be the main one that we want to notice, which is all of this gray matter that wraps around the outsides. And again, on this picture, you can really nicely see the gyri and the sulci of the cerebrum. Aside from having two hemispheres, the cerebellum is going to be its own structure. But the brainstem is made up of three other structures, namely the midbrain, the pons, and the medulla oblongata. As the name suggests, the brainstem projects out from the brain and gives rise to the spinal cord. The functions of the components of the brainstem are to ensure the survival of our bodies. We're going to point out the individual anatomical parts of the rest of the principal parts of the brain, and the next one that we're going to discuss is the diencephalon. The diencephalon contains everything thalamus. You'll find the thalamus shown here in the center, the hypothalamus, which can be found just below that, and the epithalamus, which can be found to the top and towards the backside of the thalamus. I've already told you a little trick about the diencephalon. Everything thalamus will be included in the diencephalon. So every component has thalamus in its name. Our thalamus can be found right here in the center as a reference point, and just below it we'll find the hypothalamus. Hypo means low, so that indicates that our hypothalamus is going to be below the thalamus. And epi means upon, so our epithalamus will sit upon the thalamus, positioned slightly towards the back side. The only thing that you'll need to understand about the epithalamus is that it contains what's known as the pineal gland, which is one of our endocrine organs that secretes the hormone melatonin. Melatonin will help to regulate your sleep-wake cycles. Once we have learned these three principal parts, we can now elaborate on all of the other structures that are attached to the diencephalon in some way. Again, you can see your thalamus shown here in blue, and we'll also find the third ventricle containing the same space. What this tells us is that the diencephalon contains the third ventricle. So there will be this space containing cerebrospinal fluid within the diencephalon. Up here we'll find our epithalamus and the pineal gland, which can be found on the back side. And here, shown in purple, we'll have our hypothalamus that projects down from the thalamus. Now the hypothalamus is our body's major endocrine and autonomic regulator. This part of the brain controls the entire endocrine system and the entire autonomic nervous system to help maintain homeostasis. The hypothalamus controls the endocrine system through what is known as the pituitary gland. You'll see this gland shown here, and this will sit in the sella turcica of the sphenoid bone. Hopefully we can remember that bone marking from a few presentations ago. The pituitary gland will hang from the hypothalamus through a little stalk that is known as the infundibulum. And it is through this structure that the hypothalamus can communicate with the pituitary gland by releasing certain hormones that will activate or inhibit the pituitary gland in general. Moving just superior to the diencephalon, we'll find our corpus callosum, which can be shown here, our lateral ventricle, which can be seen here, and the fornix, which can be shown here. The corpus callosum contains myelinated axons that will allow for the connection between the right and the left hemispheres of the brain. The corpus callosum can be found sitting around the lateral ventricle, which can be shown right here connecting to the third ventricle through what are known as the interventricular foramen. The fornix will sit right between the lateral ventricle and all of the structures that can be found in the diencephalon. Once again, you can see the ventricular system of the brain shown on this slide. We've already seen this picture, but this lateral view gives us another idea of how these ventricles look. You can see your two lateral ventricles shown over here, which will definitely be the biggest. Through the interventricular foramen, we'll have the lateral ventricles meeting up with the third ventricle, which is roughly in the shape of the diencephalon. The reason for this is that this is contained within the diencephalon. We'll have our cerebral aqueduct, which will eventually come down here and meet with the fourth ventricle, which we'll see shortly. And then the fourth ventricle is going to be connected to the central canal of the spinal cord, which will course all the way down through the very center of the spinal cord itself. Structures known as choroid processes found up in the roof of these lateral ventricles will produce the cerebrospinal fluid that will be found contained within this ventricular system and circulating through this ventricular system. Following the third ventricle down, you can see the cerebral aqueduct shown here in light blue, and then you have the fourth ventricle, which can be found just behind the brainstem. The brainstem was made up of the midbrain, the pons, and the medulla oblongata. Those structures aren't listed on this slide, but you can see a rough arrangement of the midbrain right here, the pons right here, which has this very circular structure coming off of the front end, and then we'll have the medulla oblongata, which will be the last structure of the brain before the spinal cord is going to project. The corpora quadrigemina is a part and can be found on the back side of the midbrain. These two little bumps here are known as colliculi, and we must remember that we're in a sagittal section, so we've actually removed two colliculi off the back of the corpora quadrigemina. In fact, there are 4 colliculi, 2 superior and 2 inferior. The fourth ventricle will be found here right behind the pons and just in front of the cerebellum, and then our medulla oblongata is the most inferior part of the brainstem. The midbrain, the pons, and the medulla are the structures that make up our brainstem. One other structure that we can see up here are the mammillary bodies. These are 2 little bumps that can be found just posterior to the infundibulum of the pituitary gland. And this view gives us a better way to view the mammillary bodies found right here. These 2 bulbs here can be found just inferior to this infundibulum of the pituitary gland. So normally the pituitary gland will come out and project here, but we've removed this so that we can see a couple other structures. On this slide, we can see the first and second cranial nerves. The first cranial nerve is known as the olfactory nerve, and it will be shown right here. The olfactory nerve will enlarge at its end, where it will become what is known as the olfactory bulb, and you can see those enlargements shown right here. Our second cranial nerve is known as the optic nerve, and that will be found projecting out from what is known as the optic chiasma. We call this the optic chiasma because this is roughly positioned as a cross. And the purpose of the optic chiasma is to allow the 2 optic nerves to travel back to the occipital lobe of the brain while crossing and entering into either side of the brain. Just as the right hemisphere of the brain is controlling the left side of the body, the right optic nerve is going to bring vision information back from the right eye to the left side of the brain. On this slide, we can see many of the exterior structures on a real brain specimen. You can first see the olfactory nerves and the olfactory bulbs that will be found at each end. Here you'll have your optic chiasma and the 2 optic nerves that are going to project out and eventually attach to the eyeballs. Just behind the optic chiasma, we'll have the infundibulum, and behind this, we'll have the 2 mammillary bodies. Your cerebellum will be found here with its 2 hemispheres, and then you'll have your pons jutting outwards from the brainstem, your medulla, which is going to be the last part of the brainstem, and then the spinal cord that is going to be attaching to that structure. Here you can see a sagittal view of the brain with the principal parts labeled and some of their components. Our 4 principal parts of the brain are the cerebrum, the brainstem, the diencephalon, and the cerebellum. You can see the cerebrum shown up here with its gyri and sulci, and it will also have fissures, which in fact we are looking at right here. Since we've taken a sagittal section, we've had to slice the brain down the center, and we're actually looking at the longitudinal fissure right here. We'll also find the cerebellum, which can be found inferior to the cerebrum, and we'll have the diencephalon, which can be found in the center of the brain. The brainstem and its individual parts can be shown right here. We have the midbrain as well as the corpora quadrigemina on the back side. We have the pons shown here with the 4th ventricle found right behind it. And then the medulla oblongata shown here that's going to project into the spinal cord. The fornix of the cerebrum can be found right here, as well as the corpus callosum, which can be shown here, wrapping around the lateral ventricles. This structure will be labeled on the next slide. We'll find the thalamus shown here right in the center of the brain, the epithalamus lying upon the thalamus, and then the hypothalamus shown down here, which is going to project the infundibulum and the pituitary gland. You've got your first and second lateral ventricles shown here, the 3rd ventricle, which can be found housed within the diencephalon, and then you'll find your 4th ventricle, which is found just behind the pons and right in front of the cerebellum. So that will pretty much wrap up the brain anatomy, but there are 3 other layers that wrap around the brain known as the meninges. The meninges are protective connective tissue layers that can be found around the brain that offer it protection. These will specifically lie between the bones of the skull and the brain tissue itself. There are 3 layers of the meninges known as the dura mater, the arachnoid mater, and the pia mater. The dura mater is the thickest outermost layer of the meninges, and it will be composed of a periosteal layer and a meningeal layer as well. The arachnoid mater can be shown here in gray, which is going to be found under the dura mater. The arachnoid mater is named because it has many web-like projections that are going to be connected to the third layer of the meninges. The pia mater is the innermost layer of our meninges, and it's also going to be the thinnest layer. The pia mater in particular will actually contact the brain itself and follow the gyri and sulci of the cerebrum. This picture will also depict the three layers of the meninges. You have the dura mater shown here in brown, you have the arachnoid mater shown here in purple, and the pia mater that can be found here in blue, coursing along the actual brain tissue and following the gyri and sulci pattern. Now, one thing that you can notice from this picture is that we're actually looking at the longitudinal fissure. And when we get to the longitudinal fissure, the meninges are going to do something rather interesting. The meninges wrapping around the left hemisphere are going to dive into the longitudinal fissure, as well as the meninges around the right hemisphere of the brain. Specifically, the dura mater of each hemisphere are going to come together and form what are known as dural septa. Dural septa can also be referred to as infolds or extensions of the dura mater as well. But we call these dural septa because they partition different hemispheres of the brain. In particular, there are three specific folds that will separate specific hemispheres. The first one that we're going to see is the falx cerebri. The falx cerebri is formed by two layers of the dura mater coming together and folding into the longitudinal fissure. This will separate the right and left hemispheres of the cerebrum in particular. That's why we call this the falx cerebri. We'll also see the falx cerebelli, which will do the exact same thing as the falx cerebri, but it will do this for the two cerebellar hemispheres. So this is going to partition the right cerebellar hemisphere from the left cerebellar hemisphere. And lastly here, we will have the cerebellar tentorium, which will dive into the transverse fissures and separate the cerebrum from the cerebellum. Here is another depiction to show you these three dural folds. The falx cerebri will be positioned here in the longitudinal fissure, the cerebellar tentorium will be positioned in the two transverse fissures, and then we'll have this falx cerebelli separating our two cerebellar hemispheres. So to finish up the brain, we're going to quickly discuss some of the cranial nerves. There are 12 cranial nerves that will course out from the brain and travel throughout the head and the face, providing sensory and motor information to different areas. Some cranial nerves will only carry sensory information, some cranial nerves will carry only motor information, and some cranial nerves will carry both. In particular, we've already seen the first two cranial nerves when we were identifying structures on the brain. Cranial nerve number 1 is known as the olfactory nerve, and cranial nerve number 2 is known as the optic nerve. The olfactory nerve is cranial nerve number 1, and it can be found enlarging to become the olfactory bulb. This nerve in particular is a sensory nerve that's going to bring information related to smell into the brain so that we can experience that sensation. Cranial nerve number 2 was the optic nerve, which is going to bring vision information into the brain from the eyeballs. So we've already discussed these two cranial nerves in particular, and we've even already identified where those nerves can be found on the brain. The last cranial nerve that we're going to specify is known as the vestibulocochlear nerve, and this nerve is cranial nerve number 8. The vestibulocochlear nerve is going to take hearing and balance information from the inner ear back to the brain. This nerve in particular is coursing out to the ear, and the bony labyrinth inside the ear has one part that's specific for bringing in hearing information back to the brain, and the other parts of the bony labyrinth will be used for balance and equilibrium. Here on this slide, you can once again see the olfactory nerves with the olfactory bulb at the end. We'll have the two optic nerves that will branch out from the optic chiasma, and down here we'll see the 8th cranial nerve, which is our vestibulocochlear nerve. As I mentioned on the previous slides, there are 12 cranial nerves. Some will offer sensory innervation, some will offer motor innervation, and some will offer both. You will not need to worry about identifying and describing each of these 12 cranial nerves for my lab class. But I have posted these here so that you can see what innervation each of these nerves carry, what the names of each of the 12 cranial nerves are, and a little mnemonic that can help you remember the order from 1 to 12. This slide will just be a depiction to show you exactly where each of these cranial nerves can be found at, branching off from the brain.